see here. So today we're talking about gene expression. You should be able to describe ways which cells react to the environment and express genes differently. So what we're going to see is a response to either external conditions, internal conditions, and we get differential gene expression. So throughout this video there will be a few videos. I'm going to pause it so you can click on the link and watch it instead of recording it, re-recording somebody else's work and taking credit for that. So I want you to do that. So that'll all be linked within the Google Classroom assignment. All right, so first thing, like I said, it's a response to the environment. So let's say you are a eukaryote and you need to break down lactose because you need the energy from that, and in order to do that you need lactase. So you only need to build lactase to break down lactose if lactose is present. What we have here is the process of transcription. So remember, in order to start transcription you need a promoter. Here I have the promoter that's gone onto this piece of DNA, and it's going to transcribe that DNA to make some mRNA to make a protein. And it's going to do that in response to an environmental cue. Here we're talking about the operator and operon. So here we have the promoter where transcription starts, and we have this little kind of key right here. And so when that is free, when tryptophan is absent, the repressor is inactive and the operator's on. So here we have the transcript being made because nothing's in that operator. Now, if this changes shape, it's going to fit in there and stop it. So the trip operon, here we have the DNA, here we have the mRNA transcript, this is the protein. If tryptophan is present, it's going to pop into there, and it's going to block the promoter from starting, and we're not going to make RNA, which is fine because what's that saying is I've got tryptophan, I don't need more, so you don't need to make more. If it's absent, then there's that conformational change, and it changes shape and it's going to come out, and then we do make the transcript so that we can make tryptophan. So repressor, this is a repressor because it's stopping. A repressor inhibits transcription when tryptophan, which is the co-repressor, is present. This is kind of like what we had with our allosteric reactions with enzymes. Co-repressor is absent, the repressor is inactive, and transcription occurs, just like we're seeing here. There's no tryptophan, it changes shape, it's not fitting into the repressor, and we start. So what is meant by a conformational change? Remember, a conformational change is when one piece fits into another that changes the shape and therefore changes the function. What type of feedback loop is seen in the example of the trip operon? So if I go back, I've got the picture of a thermostat. This is a negative feedback loop. So when it gets too hot, it cools down. When it gets too cool, it heats back up. It goes the opposite way of what was happening on its own. That's a negative feedback loop. When the co-repressor binds with the repressor, what happens to the transcription for the tryptophan gene? Double check to make sure. So it's going to fit into our DNA and it's going to stop transcription. Alright, so the lac operon is kind of the opposite example. Instead of a negative feedback, we have a positive feedback loop. I know you guys haven't been to too many rallies, but when you get to the next one, I guarantee it's going to happen. Somebody with the mic is going to get too close to the speaker, and you'll get that harsh, awful feedback sound. That's a positive feedback loop. So here, if we are dealing with lactose, when do we need to make lactase? When we have more lactose. So the more lactose, the more lactase that's going to be made. So this is going to be an example of a positive feedback loop. So when lactose is not present, the bacteria does not have to produce lactase to metabolize lactose. So we have this operator right here. It's plugged up by the repressor, all right? And so transcription is not allowed to happen. But if we get lactose, the lactose makes its way in, and its conformational change comes out of the operator, and transcription is able to happen. So instead of calling this a co-repressor, it's called an inducer because it's inducing the transcription. So how is the lac operon different than the trip operon? Remember, I said they're kind of like opposites. Positive gene regulation. Bacteria prefer to use glucose over lactose. Often both are present. How does the cell know whether to transcribe the enzyme galactase or not? So I've got a lot of glucose here, and I need galactase to deal with that glucose. Let's take a look at the animation. And it's not working. I'll go ahead and pause this, and we'll see it. It's like we said, sometimes we need to break down a molecule so we can get glucose. If we have a lot of glucose, we don't need to break that down. We don't need the galactase. So if we take a look here, maximum transcription of the lac operon occurs only when glucose is absent. All right, so we don't have glucose seen here, so we're going to transcribe to make that. And notice cyclic AMP is acting like an inducer. It's fitting into this activator protein, kind of like the operon. And so take a look. We have glucose right here. As those disappear, now we start to make cyclic AMP, the camp. Now that we have extra, they're going to fit into this guy right here. It's going to join on as the operator, and then the promoter will work, and then we're going to transcribe and make the transcription, the mRNA, so that we can make more galactase to make more glucose. And once we get more glucose, then this will stop. All right. So I'll go back, watch that one more time for you. Oh, maybe not. Here we go, one more time. No glucose, so now it fits on, and we can transcribe. All right, so what will become of abundant when lactose is metabolized? So if I break down lactose with galactase, we get glucose. All right, so differential gene expression in eukaryotes. So I've got a signal, some sort of signal. It could be a ligand, it could be UV radiation, whatever it is, there's some sort of stimulus. That leads to chromatin modification, which leads to opening of our DNA so that we can transcribe it. Remember, the exons exit the nucleus, the introns stay behind. So 20% of our genes are expressed at any given time in eukaryotic cell. Why is that? Why is it only just 20%? So a big part of the reason is all of your cells are slightly different, but they have the DNA to make everything. So a muscle cell is different than a red blood cell, which is different than a neuron. So they're going to make different proteins. They're going to activate different parts of the DNA. So they don't need to activate everything, just the parts to do their job. So that's why only 20% of those genes get expressed in any given DNA. So just a review of chromatin. Remember, we've got our chromosome is the tightly coiled, but you can't really do any work, can't produce mRNA that way. Chromatin is just the thin spindles, and that we want to be able to access. These little guys are our nucleosomes, and what we have are little histones, which are proteins, which the chromatin wraps around. So go ahead, take a look at this little video. It's going to show how that when you add a methyl group, it deacetylates, and that opens it up so that we can see the DNA so it can be transcribed. Right? So histone acetylation, deacetylation. When histones are acetylated, DNA can be transcribed. How does DNA methylation affect? So what's going to happen is this methylation, you add a methyl group and it's going to deacetylate it and everything winds up. And so again, watch this little video. Like I said, just go to document and click on it. I don't want to have you sit through here, me get the view and not the person who created the material. All right? Epigenetics is all about how our DNA may act differently based on environmental conditions. So in our question, how do two mice look different even though they're exact clones of one another, a big part of it is going to be this epigenetics. Your DNA will express or not express based on different environmental tags that get left behind. And we're finding that those environmental tags can even be passed down from generation to generation. So blank is like a switch and something is like a knob. So we've got methylation and acetylation, which goes where? Talk to your group about that one. It was in the little video, little quote that he made. So what are two factors that can affect your epigenome? We talked about diet, stress, UV light, carcinogens, activity, all sorts of things can affect what gets tagged onto that genome. Right. Here's another little one about some twin studies. It talks about the Goody mouse in particular. It's a good one. So review, in what ways do brown and yellow mice show in the video different? Why is that so? That's an important concept. Explain why DNA methylation and how does this process influence cellular function. Explain how two genetically identical twins are not really identical. So they may have an identical genome, but they may not have an identical epigenome. And explain why DNA from an oldest set of twin differs more significantly than DNA of a younger twin. Obviously, that's going to have to do with them having more life experience, so you add more tags. Right? This is about something really great called RNA interferon, which is an evolutionary response to help us avoid viral infections, but we've learned about how that also controls what's expressed or not expressed through some accidental work with genetics to try and engineer some flowers. Right? Review these questions, make sure you understand this. And then, so now I have this idea of turning on genes, another video. Go ahead and watch this one. Talks about how you can turn on whole segments. So in front of many genes, there's like an on and off signal. And that's going to be really helpful for us to understand with evolution. Review these. Again, we said junk DNA. We're learning that more and more of it is not junk, but there still is DNA that doesn't code for making proteins or anything. We think a lot of it has to do with things that are left over from viruses. And always interesting, cancer. So we talked about the p53 gene before, but how we turn off and control the cell cycle. And if you've edited that and it gets expressed, you're going to run into making more and more cells when you don't want them to. And finally, we have one more video. We'll watch that one in just a second, about how UV's influence on changing our DNA, leading to cancer, those sorts of things. All right. So, I hope that was helpful. Go ahead and add those to your notes.